you for joining us for another episode of Max Planck Florida's Neurotransmissions podcast. Today's guest is Dr. Kuan Hong Wang, who is a professor from the University of Rochester Medical Center. Throughout his career, Dr. Wang has been focused on understanding increasingly complex questions about how circuits develop and are shaped by experience. And he's also been at the forefront of in vivo approaches to address these questions. In this episode, we discuss how Dr. Wang's philosophy has guided his research interests and, in turn, his career path. We also discuss his role as co-director of the New Brain Aging Center at Rochester, which focuses on emotional well-being and its role in health, aging, and disease. Dr. Wang, thank you so much for coming down to visit us here at MPFI. We're thrilled to have the opportunity to sit down and talk with you today. So thank you for joining us on the podcast.
Yeah, it's my pleasure to join this.
One thing that really struck me in looking at sort of your research program is that you're making these, impactful contributions to what appears, at least to me, to be sort of a wide diversity of topics, from basic research into like top-down control of sensory motor function, developmental plasticity, and cognitive functioning, as well as more translational aspects. So looking for biomarkers in human disease, and now most recently sort of emotional well-being and complex topics like that. So I'm wondering, what is sort of your approach to choosing which questions you address? And are these really diverse topics, or is there sort of a common thread that unites them?
Yeah, a great question. So I think, there's maybe, from my, career past, how I get into science, I guess, when I was a kid, in high school or in college, of course, we think about this wild dreams, how the brain works, how the brain develops. So at that time, I was thinking a lot of these wild, open-ended questions. But once I started to try to really learn science, to find out how to do it, I think, I need to pick something trackable. And that's why I think my career and my interest is kind of just follow that path. When I was in undergraduate, when I started, I really trying to learning the basic biochemistry, signal transduction, hardcore protein chemistry. Then when I was in graduate school, I think, while I learn this biochemistry, I want to start look at some molecular cues important for the neural cell biology and development, neural development. So I go into that round. Then after I finished my PhD on studying the molecular regulation of exon guidance branching, I think, I need to know more about the function of it. So then I started in my postdoc to start looking at a more neurocircuit system level question. And when I go there, I think I need to start from something that has a solid foundation. So I start with the visual system, the classical, but then applying to the, at that time, the more advanced in vivo imaging, genetic tools. Then once I have the opportunity to start my own lab, I decided to take again one step further to not only looking at a more well-studied sensory system, now more going to the frontal, top-down motor control to cognitive control, and then after I moved to University of Rochester from NIH, I have the being in an so-called extramural scientific environment, I have a lot more freedom. So I basically just keep developing, advancing my, basically trying to reach bigger and bigger application and significance now that involve not only the cognitive part, but also the emotional part of the behavior and what a neural mechanism underlying this, and how that may be directly linked to the translational human clinical studies. So I kind of grow my research interest from a very focused point and then kind of expanding it. But even when I reach more recently, more this kind of psychological or human translational, I always try to ground it into some tractable tools. So I'm now just studying it at a psychological construct level, because I'm not a psychologist. So I actually want to find a bridge that I can communicate with the people in different disciplines and help to bring some of the higher level concept and questions to some more tractable brain system circuit or whenever possible, concrete molecular tools to help us address those questions. Yeah.
Yeah, so it's interesting that you mention working with people across disciplines, because I noticed that was also sort of a common thread in your career.
Yeah.
And I'm wondering, are there any like challenges that you have in working with collaborators from such diverse perspectives of science?
Yes, totally. I think for the interdisciplinary collaboration, on one hand, it's totally stimulating. This is the great opportunity that you can bring people from different perspectives together. You can have a kind of a synergistic conversation and discover new things you never thought about before. But at the same time, could be a lot of translation problem, and even the same words could mean completely different meaning in different field, or even you think you're saying one thing, but people may be interpreting quite differently. So to engage in this cross-disciplinary interaction, it's really require people from both all the sides involved to have to be actually also to be both creative and humble. Just keep asking really naive questions. What do you really mean by this word, and how would you define it? So kind of having this attitude is always open-minded, willing to learn, and so then people can truly understand each other. It's not about which, it's not about one field is teaching the other field. What's the better way to do it. It's really trying to find a common language and listen to each other. So I think so that's not only the scientific part. There's also kind of the personality part and how you can effectively communicate. So that takes efforts, and not everyone enjoy doing this. So
your career trajectory started at like single molecules and has sort of grown into these complex systems and higher emotional orders. What are the sort of lessons that you've learned along the way that sort of inform your approach as you start taking on these more complex topics? And how might that differ from like a psychologist that might be approaching things from a top-down approach?
Yeah, that's a great question. Yeah, I also wonder that some I guess maybe from my own training, I can see more people or from reductionist then go up, trying to integrate more. It seems to be a little bit harder if you start with a very high concept and then try to drill down. I wonder, this is totally my personal opinion. I wonder whether that is, maybe it's built into our brain, our human nature. It's just, and also how we learn things as we age. We learn. Typically, many people learn very well from examples. You have a concrete topic, you can really, become the expert in it. Then you have the bandwidth to gradually grow, and then you can try to approach bigger and bigger psychological concepts, for example. But if you start at that level, it's often difficult, I think, for people to drill down. And I think especially some of the technical aspect, I would think that as people age, it's become harder for them to drive in. So I think it's interesting. I think it seems to be more common to see people going this way. Yeah, to some extent, I would say I'm also inspired by my postdoctoral mentor, Dr. Susumu Tanigawa. He's a great Nobel laureate, and he throughout his career he also switched field several times. He got Nobel Prize for immunology, but he started as a pure molecular biologist. In the early days of molecular biology, working on the virus, phage, And the immunology is already, he made a drastic departure when he opened his own lab, leave the traditional molecular biology field and enter the immunology, and he addressed this VTJ antibody, gene recombination. Then after that, he made another kind of a complete different jump to the neuroscience, and then, he is actually, not just tackling the basic synaptic physiology, but he's actually asking the bold concept, what is memory, what's engram. So to some extent, I'm totally inspired by him, and I actually remember at one time he was telling me that it's healthy to switch your field every 10 years or so. but practically it's very hard, right? So once you need to be willing to take hits and willing to learn completely new things if you want to go into new areas. It will basically not afraid of going out and reaching out and not just staying in the comfort zone.
Yeah, that's really inspiring. And it seems like you have had a very open mindset throughout your career and been willing to try new things, particularly. I'm thinking also, in addition to your scientific questions, your technological development. So you mentioned, your work in Tanagawa's lab. There you developed a genetic animal model that you could use not just to answer the initial question you were interested in, but has been, I think, useful sort of throughout your career to address a lot of different questions. And maybe we could actually go into a little bit of detail about, that tool and how it's sort of opened up a lot of different directions for you.
Right. Yeah. So at that time, those were the now it's already kind of early. Going back now, those are the early days when people just started thinking about using immediate early genes to report neuronal activity. There was initially a lot of pushback from neuroscientists because they think immediate early gene is firstly discovered by molecular neurobiologists, changing, but it may not be tightly linked to the neural activity. So the initial question is always about, is it just about some general metabolic changes? How specific, it can tell us about the neural architecture and neuronal activity.
These immediate early genes, they're genes that are what happens, or what are they actually showing you?
Right. So immediate early genes are the genes that the cells will quickly respond to some changes in external stimuli, whether neural activity changes or could be other type of cellular signal changes. So the cells will show up this very rapid transcriptional response, and the new genes are produced. Now we know a lot more about those genes. They actually serve many important functions, including plasticity, learning memory, stress response, and many possible and important functions. But the early days, people don't know the functions, and a lot of question is, is it just some general marker, or it is telling us something about the neuronal activity patterns or neural representations? So at that time, I designed. Those are the early days. I designed a new mouse genetic line, put a GIP fluorescent reporter under the control of this immediate early gene, and then combined with in vivo 2-photon microscopy. So now we can see in the same animal repeatedly over time that how different, whether it's the same cells or different cells that are responding to a variety of stimuli, starting from the very basic, just the visual stimuli. So that actually demonstrates that those transcription response are highly selective. It's not just each time you stimulate the brain, all the cells will respond. There's actually very intricate patterns that actually can provide a lot of information that kind of complement the neurophysiological studies. So I think that just there are a lot of follow-up study using this concept and combined even then later the optogenetic tools. It's really becomes a very standard the neurobiological tools using this to map the circuits. Of course, there are newer tools that can get better spatial temporal regulation, but I think is kind of the initial efforts to try to build in this using molecular genetic tools to read out how the neuron functions and how the brain architectures are selectively built.
So during your talk yesterday, you actually told us about this really interesting finding looking at two different genetic models and how sort of acute changes in neurochemicals during adolescence could actually recover loss of cognitive function in these genetic models. And I would love to sort of dive into that a little bit more and talk about that study because I think it has some really interesting implications.
Yeah, so this line of work has been a continuous thread in my lab for many years. So it's all initially that there's a great kind of translational motivation. So because we started looking at adolescence when we just first started. Adolescent in mice. That is a very little talk about area. But why we care about this period? At that time I was at NIH. I've been talking to psychiatrists. They introduced me to all these neuropsychiatric disorders, and a major feature is many of them has the adolescent onset, such as schizophrenia, ADHD. All these have a very distinct temporal or developmental profile. So that kind of makes the question, what is so unique about adolescence, especially those psychiatric disorders affect some higher level cognitive and emotional control functions. So many pioneering studies from many scientists, neuroscientists have revealed that this frontal cortex is our brain's executive control center, play a major role. And then the adolescence is an important period that for the frontal cortical function development. So that lets us to see at a more kind of circuit and cellular level, what makes the frontal cortex vulnerable or malleable to the adolescent experience. So we're starting from this mouse paradigm. We just just unexpected. We find that even this simple behavior like a wheel running, just the mice like to run on a wheel, that actually can stimulate the axon growth. Particularly, this is this so-called dopaminergic neuromodulator, which are important for motivation, reward, and cognitive control. So that kind of started a series of study along this line. So we found that our adolescent experience can promote it, and we can also do a controlled experiment directly manipulating dopamine neuron activity. That will promote functional innervation, enhance circuit functions, and fascinatingly, this is very selective for the adolescent window. If the same manipulation or same behavior experience occur in the adults, not much would happen in the frontal cortex. And then we have additional conducted looking at several genetic models of which carry mutations in some critical genes involved in the synaptic functions, and found that they also can lead to this frontal cortical deficiency, which is at least a parallel to what has been observed in schizophrenia patients by the human brain imaging study. There's a reduced dopaminergic transmission in the frontal cortex. Then we try to bring this idea together. So if adolescence activity or exercise can promote it, can we use that to use this behavioral or brain stimulation treatment strategy? Can we reverse innate genetic deficits that's due to mutations? And that is also kind of came to our delight, and actually somewhat surprised, because initially we say, okay, we just do a few days of or a few episodes of stimulation in adolescence and wait a couple months and to see whether there's a long-lasting effect. It seems kind of crazy because it's a long-term experiment, and it's although there's no precedence whether this will the likelihood of this work. So it turned out after the people in my lab work really hard to try this out. And we actually get a robust effect, and we did a lot of controls so it's actually truly effective, and it's also, but of course, it's not a magic bullet. So there are a specific dosage, specific developmental window, and there's, the stimulation has to occur in a proper range. If you stimulate too much, just as we know, too much dopamine could also be bad for you. So this is a lot of implication. We actually continue studying this. A major challenge, and we're addressing now, because our previous study is all showing this adolescent plasticity and the potential therapeutic potential. But if we really want to translate into, say, the human treatment, it's a lot harder to convince people to do anything before they have a clear symptom. So whenever people really seek out treatment, it's often much later, would be in adult. But we also said that in the adult, it's very difficult to induce this plasticity. So, from this basic animal study, we actually find that a combination of neurostimulation and pharmacological treatment that actually can reopen its plasticity. And particularly this pharmacological treatment is actually a key component, a key target of the typical antipsychotic. Doctors prescribe antipsychotics to reduce psychotic symptoms, but typically that is not effective to reversing the cognitive deficit. It just helps to prevent this extreme psychosis. So we found that if we combine both the drug treatment and a properly timed neurostimulation, then we can re-engage or reopen the plasticity, and leads to the functional enhancement. So we are testing more along this line, including identified additional cellular players in this plasticity, as well as to find more less invasive approaches to induce the same type of plasticity. So so this is an area that we are really passionate about. Yeah.
That's really exciting, and very exciting implications as well. Mm-hmm. I'm curious about the circuit mechanisms of it. Do you know which circuits are involved, and is it really circuit remapping that's going on?
Yeah, so at the circuit level, also what we identified, the immediate, very specific change is the neuronal projections from the dopamine neurons in the midbrain VTA area to the frontal cortex. This plasticity, just the first sign is increasing the axonal boutons appearing on them. So the recent studies, we actually we're probably going to submit a paper very soon. We identified additional non-neuronal contribution, so the local microglia contributing to this plasticity, and it also have a very unique developmental time window. So so this is also, it's not just about a particular neuron-to-neuron interaction, but the other cell types in the brain also involved.
This new NIH initiative that you're part of. So you're one of the co-directors of the new brain aging center at Rochester, which is part of the Network for Emotional Wellbeing. Could you tell us a little bit about what the goal is? And you're one of 6 different centers.
Right.
And what the differences are between the different centers.
Yeah. So this is actually this is a very, now we're moving to a very high level concept. The emotional well-being. This is the kind of the word that everybody can relate, but the people ask what it is and how do you study it. So there's first of all, this is originated from some workshop a few years ago. NIH and also a Surgeon General of the U.S. They had organized a workshop in response to a big needs to understand well-being and what are the different components of well-being, physical, psychological, and then about psychological, what are different components, and then the NIH created this call to basically to ask the researchers, form these different networks in order. So the purpose of this network is actually because it's really a new field to have research coming together to discuss, even including discuss a definition, how do you measure it, and whether it's possible, how to go to the mechanistic level. So the different centers across the nation, the six, we all have a different emphasis. Some is more on the peripheral kind of the biomarker, physiological markers. Some are more on the central or brain imaging part, and some will focus on the measurement in the developing children. Like our center is focused on both the brain mechanism as well as the implication of aging, because from the human studies, there's a really fascinating observation as people age. They actually generally, even though there may be physical health decline, but they often take a more positive attitude, from their emotional perspective. So, what could be the mechanism? How many of that is a kind of social, or how many of that could be truly due to the biological changes? So there are a lot of really open questions there, and from my role and my perspective, I'm also trying to kind of to represent more like a neuroscientist view on how to approach these questions, and how this kind of very high level concept, how we can dissect into individual components. For example, to what extent that this well-being is experienced at the moment, so which will be more like a affective neuroscience, that just how individuals perceive positive and negative stimuli differently. And then what is the evaluative or reflective component? It's just how individuals using cognitive functions to make judgments over a longer period of time, whether this experience is considered adaptive or maladaptive. So we're trying to create dialogues among both the human clinicals and the human scientists with the basic scientists, and to define the tractable components of this high-level psychological construct, and then also develop circuit, or even possibly the molecular mechanisms into this. So even though this is a very high-level concept, but we are studying, for example, at the molecular level, we're studying the cannabinoid system and the dopamine system. So we all know that when how drugs affecting this pathway will change our perception, emotional processing, and also how we evaluate the environment, and so this is kind of seems there's a fundamental component. But neurobiological components underlying this higher level concept. So this is really want to, through this initiative, to encourage more dialogues and gradually and actually better define these concepts. Yeah.
And reading through some of the papers you've put out on this topic, it seems like a real focus is not just on abnormal aging, but also on traditional aging and quote unquote normal aging. Is that fair to characterize? The goals?
Yeah, so I guess this is both interested in the normal aging as well as to the implications to the pathological, in the dementia, the AD, Alzheimer's disease situation, because it has been recognized. Even though the Alzheimer's disease is commonly the first thing jump to people's mind is the dementia or loss of episodic memory. But the development of psychiatric symptoms, is actually both it can predict the progression of the cognitive symptom. So there's actually a ongoing research, whether the psychiatric symptoms may directly exacerbate the cognitive decline rather than just a simple or parallel readout. So basically, to better understand what contributes to the emotional well-being is not only just promoting health under the normal during normal aging, but it could also have great implications in potentially prevent or at least slow down the progression of neurodegenerative disorders.
What do you think the biggest gaps are in our knowledge to really be able to understand a concept like emotional well-being in terms of neuroscientific perspective?
Yeah, I think that because this is still a very psychologist-driven concept. So it's just the bigger challenge is to come up with some measurements. So yes, we can talk about it, but to go after scientific inquiry, what set of measurements will be useful to read out whatever animal research experimental manipulation can be introduced. So this is a ongoing challenge. It's not just it's also just for the concept of emotion alone. So, there's a rich history of debate. What is emotion, and there are actually completely opposite views. Some people feel like emotion is totally human-centric thing, and then there's the other spectrum is actually even originating from Charles Darwin. It's just emotion is how animals motivate themselves to adapt to the environment. That's the fundamental function of emotion. So in that case, it's not just restricted to the human. So I think from there's a lot of exciting recent progress in emotion research. The idea is the human research, of course, is essential. You have the subjective report that will map onto what everyone can agree on what is emotion. But at the same time, through the brain imaging or the autonomic or physiological behavior measurements, those are more objectively measurable markers. So the challenge for the translation or research is to find the appropriate markers in the animals that is relevant, is logically relevant to the particular species that you study, but have a common thread, even linking across species. Then, if we can identify that kind of markers, then it will make the animal research, the targets will be a lot more clear. So this is really kind of a constant dialogue between the animal and the human research. And the human research can find the correspondence between the subjective report and what kind of the objective marker. Then the animal research can take this objective marker, then do perturbation studies to find what kind of which brain part, which circuits actually will affect this objective marker. Then hopefully some of this manipulation can be delivered relatively non-invasively or through some kind of a cognitive behavior therapy that we can human to train to engage those circuits. That will complete the full circle. I think that at the early stage is really just to bring people together, willing to discuss this, and not just treating them as a completely different realm of research. That is already that's actually all these research networks NIH funded. That's what we are trying to achieve as a first step. Yeah.
I wonder about your perspective on looking sort of in the field of neuroscience as a whole, and maybe even more than neuroscience. Mm-hmm. Do you see these large network type research programs as sort of the future to tackling these more complex and abstract topics?
Yeah, I think it's this is a complex topic, but I would think that science always benefit a kind of a more diversified view. So depending on the progress of science, both individual investigator-driven kind of exploration as well as more systematic organization, either through a network or even more aggregated into some kind of center or central institute. So I think that will map onto different questions. I would say in a field that people don't really have a clear, we don't even know what we don't know. So in that consideration, definitely individual, it will be the individual talents and the exploration that will benefit the science growth. If it's too top-down control, then if one central person make a mistake, then it will push the whole research field in the wrong direction. We don't want that. But as the particular question become more and more mature, then if you have a very well defined to the extreme like an engineering target, then it totally makes sense to build up a standard operation procedures. So when you get more kind of engineering application or clinical application, you want to standardize that so that it can be done efficiently and the goals can be achieved in a timely manner. So I think it's really depending on the specific questions and the stage of the scientific development.
Well, Dr. Wang, thank you so much for sharing your expertise and your perspectives with us. It's been a pleasure talking to you.
Yeah, it's my pleasure talking to both of you. Thank you so much for giving me this opportunity to talk to you.
Thank you.
As always, thank you for listening to Max Planck Florida's Neurotransmissions podcast.
If you enjoyed this episode and would like to help support the podcast, please subscribe and tell your friends.